Okay, thank you. Good morning, everyone. Thank you for the introduction and for the opportunity to present the work from my PhD. So if I start with a brief overview to the biology of the system that our lab works on. So if you imagine a pathogen invading you or a host, there are two main types of immune response that are mounted. These are the innate and the adaptive immune pathway. Our lab actually works on a third immune pathway called the complement system, which kind of bridges the gap between these two pathways, and it's an entirely protein-based immune pathway. And it has many functions within the cell, but the one that we're most interested in is the pore formation and regulation of a beta-barrel immune pore called the membrane attack complex, or MAC. And the MAC is a really great killing machine, and it can kill this pathogen that's invading you. Now, the way that this works is that these soluble complement proteins associate with each other and the membrane in this like stepwise pathway that forms this ultimate beta-barrel pore. And to do this, they have to undergo a really major conformational change. So they start in this soluble conformation, and the hydrophobic membrane-spanning region is hidden away in these helical bundles. And then upon membrane insertion, this unfurling event happens, and they become beta-stranded. And it's in this beta-stranded conformation that the full pore can polymerize and form this beta-barrel immune pore, which was solved by the Bubek lab a couple of years ago now. And so this, as I mentioned before, is a really efficient killing machine, and it has the ability to kill this invading pathogen, but it's also got the ability to deposit on your healthy host tissues, which could cause kind of off-target or bystander damage. So there are a number of regulatory mechanisms to kind of overcome this, and the one that I've been looking at during my PhD is this GPI-anchored protein called CD59, which is expressed on virtually every single human cell. It's a vital immune homeostasis protein, and without it, MAC would be essentially able to kill any of the cells, any of your healthy tissues during an immune response. So what this protein does is it's anchored to the outside of the plasma membrane, and it gets in the way during the stepwise assembly pathway of MAC. And there are two possible points of control, which I've highlighted here. And essentially, what this protein does is somehow, by binding to the complex as it grows, it prevents rupture of the lipid bilayer, and it prevents C9 polymerization of the full pore. And so to try to understand this mechanism, we wanted to solve these two structures using single particle cryo-EM to understand the structural basis of this mechanism. So to do this, we needed to find an efficient way of inhibiting MAC. And as I mentioned, CD59 is actually a GPI-anchored protein. They're quite difficult to extract from the membrane, but there are a number of synthetic GPI analogs that we can use to kind of capture GPI-anchored proteins on membranes synthetically. And this is super important for CD59. It's about 50 times more effective at inhibiting MAC when it's localized to the membrane. So we use this myristylated myristylated cytotopic peptide, which gets linked to the C-terminus of our protein, and then it can spontaneously incorporate into the membrane. And on the right-hand side here, I'm just showing a simple C9 polymerization experiment, which we can show that in the presence of this analog, we get inhibition of MAC. So once we'd found this method that was helping us to capture inhibited complexes, we moved to try to purify these and use them for cryo-EM. So to do this, we kind of used empty nanodiscs as a scaffold, and then by mixing together all of the soluble proteins that make up the complex and our nanodisc, we could kind of back react them together to form our inhibited MAC structure. The reaction is kind of a lot more dirty than just simply making our inhibited complexes. So we needed to purify it, obviously, and we purified it using density centrifugation because we didn't have any kind of native style purification. So I'll spare you the details of the purification, but essentially we took our gradient, screened every fraction, found the sample that we were interested in, and then concentrated it for cryo-EM. Unfortunately, we had problems with the stability of this complex. So to deal with that, we had to use a glutaraldehyde cross-linking to stabilize the complex so that we could image it. This was quite a neat method for us because we didn't have to change our purification. We just needed to add glutaraldehyde to use gradient fixation. But this significantly improved the stability of our sample in ice. And this actually worked for both of those two assemblies that I mentioned at the beginning. And so we were able to collect high resolution data sets on both of those two assemblies so that we could on both of those two assemblies. And these are just representative 2D classes and micrographs from ththose two data collections. So I'm going to talk more about this second data set because there was more heterogeneity, but both of these two were solved in a very similar way. So when we first did initial consensus refinements of this structure, we had a significant problem with density deterioration. So essentially, the complex likes to align on this section at the top, and along the arc of the pore, which is where we have most of our heterogeneity, we lose a lot of density. So, and in particular, we had absolutely no density for the inhibitor protein CD59 on this complex. So to overcome that, we did a number of stages of classification. The first stage of classification that we did was to separate these uninhibited MAC structures from our CD59 inhibited MAC structures. This was relatively simple with 3D classification. We could tell the difference between them based on the arc size of the pore, similar to that very first experiment. What was a lot more challenging was separating much more subtle stoichiometric differences within the complex. So we did this using density subtraction and focused 3D classification, but what was really important to us was about choosing which protein, which subunits were in that density subtraction so that we could separate the different stoichiometries. And then we were able to do full refinements on each of these complexes to give us the different stoichiometries of the complex. Now, I'll mention here that we don't actually see a 1C9 structure in any of our data set, and that becomes important for the mechanism, which I'll show in a few slides' time. So from these refinements, we did local resolution filtering, and although this gave us a kind of a first insight into how CD59 was binding to our complex, you can tell from these locally filtered structures that the local resolution of CD59 is pretty poor in this area. It's about 8 to 10 angstroms. And so we wanted to try to improve that so that we could unambiguously fit the crystal structure of CD59 into the density. So we did this using a similar method to what was mentioned yesterday, which is doing masked-focused 3D classification with a really high T value to separate different positions of the CD59 protein. And so this gave us one class that looked kind of well ordered and nice, and so when we refined that to high resolution, a full 3D refinement, then we significantly improved our density for CD59. But we knew that we could improve this further using, again, density subtraction and focus refinement. And by doing that, we were able to resolve the secondary structure details of the CD59 protein, which allowed us to unambiguously fit the CD59 crystal structure. And from here, we can build near complete atomic models of these 3 out of 4 of our structures, including the C5b8 structure, which is from a different data set and was actually solved by one of the other postdocs in our lab, Scott Gardner. So from here, so from here, probably the most striking thing about these, all of these different assemblies of the inhibited MAC is that CD59 binds to exactly the same protein, which is this protein in light pink, which is called C8 alpha, which is the this protein called C8 alpha. So if we look at the interaction between C8 alpha and CD59 in more detail, this is an anti-parallel beta-stranded interface between these two proteins. It's stabilized by a number of kind of salt bridges and pi stacking interactions. But the thing that's kind of what really surprised us was that there are these 3 consecutive highly conserved glycines in the middle of this interface, which are obviously not making any useful side chain based interactions between these 2 proteins. So what we think that these 3 glycines are doing is introducing flexibility into the complex that CD59 can take advantage of, and as a result allows the trajectory of this beta hairpin to be altered such that it can't productively insert into the membrane. And just to demonstrate how this flexibility is, did some 3D variability analysis, and this can tell from move the trajectory of this hair is changing quite rapidly. So we think that this is how CD59 is preventing membrane rupture by this particular protein. It's not this protein where membrane rupture is prevented. CD59 also prevents membrane rupture the next protein. What it does is it induces the really kind of drastic bending of the beta hair protein by an angle of about 6 degrees. So in this conformation, this beta hairpins are going nowhere close to the membrane, and again, it's preventing rupture of this particular rupture of the membrane protein. Now, 2 pieces of information kind of explain how CD59 is preventing membrane rupture, but they don't really explain how CD59 is preventing full polymerization pore. So to do that, we need to look at the final of each of the complexes, and this is yet another different conformation of this protein. In this particular case, we have 2 equal bundles which needs to unfurl into a beta hairpin, remain in its helical conformation. Keeping this in the helical conformation acts as a steering block on further polymerization of the pore. Now, intermediate conformation was actually first captured in a different inhibited assembly of the MAC complex by a former in the group who solved the structure on the right, and similarities between kind of that inhibited assembly and so this is kind of a mechanism for how polymerization of this particular complex is all. However, this seems to be kind of effect of CD59 and it's not directly associated with it. So those kind of answer the questions that we started the project with. We had the unique opportunity to solve this complex in a membrane environment to look at the impact this will have on the membrane. So if we apply a positive factor to our map, we can look at kind of low resolution changes are happening at the nanodisc in response to this complex binding. And so we see this kind of local thinning of the membrane around these early complement proteins. On a cellular level, this might act as a signal for lipid signaling or the recruitment of GPI and CD59. So to test this, we have done some preliminary cellular experiments. We perturb the plasma membrane using classical depletion. We can knock out the ability for the MAC proteins and to coagulate in the kind of absence of cholesterol in the membrane. And just to take that a step further, this also removes the ability for internalization and subsequent clearance of the complex from the membrane. So this is preliminary evidence that the cool changes in the membrane are impacting the regulation at the cellular level. So from here, we were able to tell a structural basis for this mechanism, where we think local changes which happen by these early complement proteins as recruitment signals. CD59 then binds to the complex and it changes trajectories of the beta hairpins as they unfold membrane rupture. And this has a knock-on effect on the complex, which means you get a stabilisation intermediate conformation of C9, which prevents further polymerization. And then most likely this complex gets internalized and cleared from the cell membrane. So with that, I want to acknowledge a number of people who were important in this work. Firstly, Dora, for having me in her lab for the last 4 years, had a really great time, and everybody else in her, particularly Scott Gardner, who I presented today, the team and who helped data in various levels of COVID lockdown, and UK and the European Research Council, who funded work at the time, and I look forward to questions.